This is module A&P, module 5, part 1. So here we are, muscle histology and physiology. So we have reached the muscles, and on page 125 introduces the whole shebang and offers some pretty little pictures at the very beginning too. So we will study through this. Was there, I went over the test just quickly, and really there wasn't much to go over, but was there any questions about module 4, anything you needed to know? Are we good? Alrighty, so here we go. Muscle histology and physiology. So again, from your terms, your notes, that's the stuff that I prefer for you to know. Muscle histology, of course, muscle tissue has 4 functional characteristics. Contractibility, in other words, the muscle can contract or shorten forcefully. Excitability, it can be stimulated by nerves and hormones. The nervous system and the endocrine system can control the muscle system, so that's how you move by virtue of those 2 things and how you survive. Extensibility, under normal conditions, muscle can be stretched out or relaxed is another way to depict that. So we are either contracting or we're sort of relaxing. And then finally, elasticity. Muscles can be extended beyond their normal length and then recoil to regular length. And of course, you don't want to do that too far because that produces something called muscle strain, which is not just I'm tired, says the muscle, but no, that is actually a tearing of muscle tissue. So that is something that we all need to be careful of. So muscle tissue performs 3 basic tasks. It provides movement for the body, that moves the skeleton and many organs in the body. And we'll get to page 126 in a moment. It maintains posture. So there you go, good posture. And without muscle activity, your body would collapse. All right, so that is the case. And then one other important, and in the wintertime especially, well, you recognize this in the summertime too when you exercise, it produces heat for the body. So when you're exercising or doing work in the summertime, you recognize that heat and you got to do something about it, cool off. But the opposite is the case in the winter. You to try to warm up in the wintertime, and you know that by using muscles, you can kind of do that. In fact, it's the oddest thing, isn't it, when weather is very cold and you've been outside doing something and you find yourself stifled and hot in even a thin jacket. There you are in a thin shirt and it's 30 degrees outside because of the ability of your body, your muscles, to produce heat and warm you. So there are 3 kinds of muscle tissue, and those are depicted in your pretty little picture there at the bottom. There's skeletal muscle that's striated in a striped pattern, and you see that depicted there. That includes voluntary muscles. Those are muscles that can be consciously controlled. Multinucleate cells, which wake up muscle tissue that is controlled autonomically by the nervous system, but can still be consciously controlled. So that is skeletal muscle. There is cardiac muscle, unique to you, found only in your heart, striated with single nuclei, is involuntary and cannot be consciously controlled. So, in those, if you've seen some of those martial arts movies, the old ones especially, I'm thinking of a martial art TV show, Kung Fu, when I was a kid. The lead character was said to have eluded someone by virtue of relaxing himself to the point where his heart slowed down to almost a stop. And the truth be told, no one can do that. You can relax to the point where your body isn't needing a lot of circulation and your heart slows, but you cannot control cardiac muscle just by virtue of your or whatever you might call it. Then there's smooth muscle, and you saw the cardiac muscle depicted there on the right. Smooth muscle is involuntary as well. It's non-striated, in other words, not striped, and this helps with organ function, and we'll look at these as we go through. So let's start with skeletal muscle. Turn over if you would. We will take a look at skeletal muscle both macroscopically and then microscopically in the little picture there, the little figure on page 126. A helpful picture for you. This probably looks a little bit familiar in terms of the nerves as well. So skeletal muscle structure is, well, it connects to bone by a tendon. So your Achilles down here, and surely hope no one has ever messed up their Achilles. Anybody here? So first year I taught A&P, it was a girl who was a senior and was going to have spit it out, Roy, soccer scholarship to Auburn. And that fall she tore her Achilles, and it was just awful. It obviously messed her up terrible, but turns out she the folks there at Auburn ended up continuing with the scholarship, for her to play. She wasn't able to play that next year. Those are very, very slow heals, but in any respect. Skeletal muscle connects bones by a tendon. That's a strap of dense, regular connective tissue. It contains many nerves and blood vessels. Muscle movement requires energy that is delivered in the form of oxygen and nutrients through the blood vessels. Movement and muscle contraction would not take place without nerve stimulus, okay? And so we know that to be the case in terms of spinal cord injury. You know that if with spinal cord being damaged or severed, you know that people are unable to move that portion of the body affected. So you have to have stimulus from nerves. So let's look at this thing, the belly. The belly of that muscle, you see it depicted on the regular sort of size, is the muscle itself, and it is composed of muscle cells. The fascicles are bundles of cells that make up the whole the whole muscle, and so what we'll be looking at increasingly in just a moment are these fascicles. All right, you see it depicted there in terms of the muscle itself, and then the breakdown. Well, in terms of the drawing, I wish I could refer you to it, but you see fascicle there to the left of the first blow up, and depicting one of those little round sausage-looking things. Is that all right? The muscle fiber or muscle cell. Each muscle cell is actually a long, thin fiber. So that is depicted both in the blow up on the right, and then of course it is ultimately blown up on the bottom in terms of each individual muscle cell. So you see it in terms of pointing to the muscle fiber within a fascicle. Then one is sort of stretched out there in the middle. And then that is depicted on the bottom. So that's what we're talking about. The endomysium is a thin collagen layer that wraps each muscle cell or fiber. These fibers reach past the end of the muscle belly. They straighten out and form dense, regular connective tissue of the tendon. So that's how the tendons are ultimately formed, which then continues into the bone and becomes part of the bone and or the bone collagen. So that's how these muscles are connected in terms of your bones, and you are ultimately able to move. So the endomysium there, you see it depicted, well, it's depicted in the first blow up there, that endomysium. And then the paramysium is a collagen layer that wraps each fascicle. So again, you see the fascicle as the first sausage, so to speak, and so the thing that wraps that sausage, that encases it, is the paramysium. So the epimysium, or the fascia, wraps together several fascicles, and it differentiates one muscle from the next. So you see the epimysium depicted there. It's the top word from the blow-up, the epimysium, and again, it wraps the whole shebang, okay? It wraps several of those sausages together. The sarcolemma, sarco means flesh or meat, is the plasma membrane of the muscle fiber or the muscle cell itself. The nuclei are located just underneath the sarcolemma. So if you would go to the second blow-up, so to speak, in terms of these muscle fibers, you see the sarcolemma is what wraps the whole thing there, the whole muscle fiber. The sarcoplasmic reticulum has tiny tubes or T-tubules that form pits in the sarcolemma, which makes it possible for messages to get where they need to be in order to stimulate the muscle movement. They surround the regions. They're packed with myofibrils, or thread-like structures that extend end to end and are made of repeating striations. Each myofibril contains two types of myofilaments, actin and myosin myofilaments. So that was a mouthful. So you see the sarcoplasmic reticulum there in terms of that depiction. That's sort of in the, I guess that's kind of a blue in the second blow up. And then the individual myofibrils are those little things that ultimately, well, they're not well displayed, but the myofibrils, each myofibril contains two types of those myofilaments. You see a myosin myofilament and an actin myofilament, and they are going to be displayed in those colors, as we will see in terms of their movement in just a little bit. But so you see how those things break out in terms of the individual muscle, and then the small portions of it in terms of its anatomy. Turn over to the next page of your notes. And then turn 2 pages in terms of how a muscle contracts to page 130. And just because, if you would, to kind of get a sense for this thing, turn back 2 pages, back to the blow-up of the skeletal muscle, and notice the colors there, the purple actin myofilament and the kind of a greenish myosin myofilament, and then compare that to what we're going to look at in terms of the movement of this thing there on page 130. So what you kind of have here on 130 is a way to try to describe this thing, and it is somewhat what it is, what it looks like in terms of it being further magnified. But that's what we're talking about in terms of these myofilaments in terms of their movement on page 130. So, what we're talking about here to summarize movement, muscle movement, is sliding filament model anatomy. All right? So that's what we're doing. We're sliding filaments. So, in terms of defining these things and how it all works, we got to define them first. And so a sarcomere, would you, again, we're looking at a single sarcomere. That's at the top of page 130, figure 5.3. So notice a single sarcomere is all that stuff in between the lines there in terms of its depicting. And that is a repeating unit of myofibril. Each unit of thick line between two thin lines represent a sarcomere. All right? So, and again, what you're being reminded of in one sense is the single sarcomere is that stuff that is depicted and we'll see in just a moment in terms of contraction and afterwards, but that's a sarcomere. So the myosin, myofilament, myo again, muscle, sin is protein. We're talking about the green is the thicker of the muscle proteins, muscle myofilaments. It's located between the actin myofilaments. It connects or touches the actin at the myosin heads. All right? So look at the word head there depicted. And so that's where the myosin is contacting the actin. It's called a head. And where do we go there? Myosin heads forming a cross bridge. A single myosin myofilament forms cross bridges with actin myofilaments from both Z discs. All right, so look at what we're defining as Z discs. So right along the side there, in terms of that actin myofilament, they then, they're displayed as they are, contact the myosin by virtue of what you see there, those cross bridges that form at the myosin heads. So, but again, in terms of the Z disc, that's what we're talking about, the little bead-looking things there on the end of each, on both sides of a single sarcomere. So the actin myofilament, or the actin, is made up of the protein actin, and again we're talking about the purple, the thinner of the myofilaments. It connects with the myosin at active sites, which become cross bridges. So again, myosin heads, active sites, they're not exactly synonymous, but they describe pretty much the same place and what's going on there. And of course, they ultimately form these things called cross bridges, where the head of the myosin grabs the actin and pulls on it. All right? So the myosin there is, in terms of it, it's grabby. All right? So take your hands off me. Well, no, if we're going to move, I've got to get a hold of you. So it grabs the actin then and pulls on it, shortening it. This happens synchronously with all of the muscle sarcomeres so that the entire muscle fiber contracts. So in terms of, again, we're talking about microscopically, we've got this grabbing going on, and ultimately the contracting then, which is done by all of these sarcomeres, and that's what will produce the contraction of just one single muscle. The Z discs in terms of these things are discs of protein that separate one sarcomere from another. The actin myofilaments are connected to the Z discs. One group of actin myofilaments stretches from one Z disc towards the opposite, which stretches from the opposite Z disc. So again, you see how that is displayed there. The actin, which connects there at the protein in the Z disc, and they're sort of heading towards one another. And you'll see how this works in just a second in terms of this grabbing on and this movement, this sliding. So the H zone, in terms of that H zone, look at the distance between those actin myofilaments, both top and bottom, but the bottom one is labeled as the H zone, and that is the area of separation in the center of the sarcomere between the opposing actin myofilaments. All right, so again, that's a label description of a place. The A band is defined by the myosin myofilament. So in terms of that myosin, you see the stretching from one end to the other of the green, and that distance, that total distance measured it has a name. It's the A band. Okay? And then finally, the I band, and that is portions of the sarcomere that includes the Z disc in which there is no myosin myofilaments. And so you see how that is labeled as well in terms of the I band from one sarcomere to the next, where there is no myosin at all. All right, so you see the I bands labeled at the end of each sarcomere. So that's a lot, but really all we're doing is identifying the microscopic portions here in terms of the movement. We haven't moved yet, all right? We're going to move, but we haven't done that. All we've done is identified the stuff involved in the movement. And so what's your job? Your job is to take a look at that picture and to be familiar with those things that we're going to describe in just a minute that will move. Is there questions about any of that? I didn't think there would be, although there should be. I've mentioned many times, I used to love micro, but for whatever reason, the microscopic portions of anatomy and physiology just don't interest me as much. I don't know why, but anyway, this is stuff we got to do anyway. All right, so take a look at page 131. There is the contraction of a sarcomere. All right. So notice the differences. That's how we're fixing to move. All right, we're fixing to contract. The heads, I'm in the middle of page 2 of your notes. During contraction, the heads of the myosin myofilament get grabby, all right, and the myosin grabs the actin myofilament and they bend, all right. So those heads of the myosin myofilament bend, and the result is they pull the Z discs towards the center of the sarcomere, reducing the length of the sarcomere. So the whole thing reduces. Look at figure 5.4. You see what's going on there. We have reduced something. Interestingly enough, notice, and I'm just going to point it out now, we're going to repeat this in a moment, but notice we haven't actually reduced the length of the A band. So in terms of the length of those myosin myofilaments to begin with, we haven't changed that. What's changing is the distance there in terms of the H zone. That's actually what's going to change. And you'll also notice, excuse me, the length of the I band. That's going to change as well in terms of that Z disc moving closer towards the myosin myofilament. Okay, so we just kind of described what's in your notes here. So we're pulling the Z discs towards the center. We're reducing the length of the sarcomere. So the length of the H zone is reduced, as are the lengths of the I bands. But three things remain the same: the lengths of the actin myofilaments, the length of the myosin myofilaments, and the overall length of the A band. So, excuse me, the actin myofilaments didn't change either. So the length of the myofilaments in general don't change, but the distance between them does. So the myosin myofilaments grab on by formation of what we've already identified, or at least we've used a word, cross bridge. There are two other proteins in the actin myofilament. Troponin, those are the brown molecules. If you would turn over, sorry about that, turn over to page 132. So there, if you don't mind, you can go back and forth. You see the little small section, which was the blow-ups of what we've just been talking about. Small section, and what they're showing you then is a blow-up of just a small portion of the actin and myosin. So in terms of of what's going on there, there are two other proteins in the actin myofilament, the troponin, and those are depicted as the brown molecules there in your figure. And the tropomyosin, which are the white and blue, and they're not, at least in my textbook, not very well depicted, but it's kind of the white and blue winding strands, the double helix actin, around the double helix actin molecules. A nerve sends a signal to the muscle fiber which causes the sarcoplasmic reticulum to release calcium ions into the sarcoplasm, which then bind to the troponin, causing it to change the way it sits on the tropomyosin, resulting in active sites which attract the heads of the myosin myofilaments, all right, and causing a binding or a cross bridge. So in terms of all of that, you see it depicted what we just read. You see the calcium ions there, and the production of that change in terms of the way these things sit, and those are depicted there. You see the troponin kind of standing up, and again, all of that to result in active sites, which then, and that's depicted from the blow-up portion there. You notice how the myosin is kind of sitting away from the actin. Well, then as a result of the development of the troponin, these active sites attract the myosin heads, and so that's when you form these cross bridges, and you see the connection of the myosin to the actin. And they result in active sites which attract the head of the myosin filament, causing a binding or a cross bridge. Without the nerves stimulating the muscle fibers to release the calcium, the muscle would never move. So last time, in our last module, we talked about the importance of calcium throughout the body in terms of both the removing of calcium from bone and the adding of calcium back into bone. And here is an example of why that's vital to you in terms of maintaining homeostasis. Without the calcium, without sufficient amounts of calcium, you ain't gonna move, all right? So that's not a hand raise. So again, without the nerves stimulating the muscle fibers to release the calcium, the muscle would never move, okay? So in terms of the cycle of sarcomere contraction, so There's a bunch in terms of this, but I told you the microscopic portions of this stuff just don't interest me as much. So what I've done is summarized what I'd like you to know. That summary is actually on page 135. There are drawings of it to remind you what's going on. Would you turn over to 135? So I have simply I have added to you the notes there from page 135 to your notes. I have just put them in there. The cycle of sarcomere contraction. This is how it all goes on. Before contraction, the active sites on the actin myofilament are not exposed. We just talked about that. The heads of the myofilament there, but it's myofilament, are ready, but there's nothing for them to do. See, they're separated in terms of of both the actin and the myosin. Number two, to start contraction, calcium ions bind to the troponin, causing the proteins in the actin myofilament to rearrange. And this exposes the active sites to which the myosin heads are attracted and then can bind. Phosphate is ejected when the heads bind to the active sites. So in terms of this process, you see the picture depicting that in number B there. The power stroke is the heads bend, pulling the actin myofilament in the direction of the bend, causing the ADP to be ejected. So in terms of the movement, you see what's happening. In fact, again, turn back 2 pages, back to the contraction of a sarcomere. Hold your finger on both, but you see what's going on. The bending is going to cause that movement. The restriction or the making it smaller, the length of the H zone and the length of the I band. Now to number 4 here, or D on page 135. The ATP molecules bind to the heads, causing them to release the active sites. The stroke from the heads has moved the actin myofilament. All right, so it's moved, but now we're going to release it, and the return stroke then allows, this is the last one, number 5, return stroke. ATP molecules break down to ADP and a phosphate, which stay bound to the myosin heads. This releases energy and allows the heads to spring back, ready to begin the process again. So that is in a nutshell, and that nutshell is what I want you to be familiar with. All right, there's more detail in here, but what I want you to be familiar with is what is in your notes, and then in terms of how it relates to the pictures. So that's a lot of stuff, a lot of little stuff, and somewhat detailed. I find that a little bit interesting. As I say, a lot of this stuff doesn't necessarily thrill me, but that's not too bad. So is there questions about any of that? Turn over one more page. I wonder if I just ought to do this next time. Let me see how much is on page 5. 4 or 5. I think I will. I think that's what we'll do. We're going to do, we'll start on page 3 next time with the neuromuscular junction. And that's kind of an interesting deal. It's a pretty little picture there on page 137. Is there any questions about the stuff that we've done so far today? This will give us time I can call your name, get you some tests back, and check your homework as well. So if there's not any questions, you guys have a great week.